We are the Gujarat Technological University. Since its inception over a decade ago, GTU has been empowering young and curating minds to realize their true potential. Over 4 lakh inspired students are enrolled with one of the premier academic universities in India. With more than 450 affiliated colleges in its fold operating across 5 zones of the state, GTU, the International Innovative University, your place to move forward.
Self Grishma Dalal from Faculty of Pharmacy, Dr. SUBHASH Technical Campus. Today we are going to focus on centrifugation as I mentioned in my last video lecture. So first of all, we are going to see the basic things about what centrifugation is. So here is the definition. Centrifugation is a unit operation employed for separating the constituent present in the dispersion with the aid of centrifugal force. I repeat, centrifugation is a unit operation that is employed for separating the constituent present in a dispersion with aid of centrifugal force. So here what are you using? You are using centrifugal force, right? Centrifugal force provides the driving force for the separation. It replaces gravitation forces operating during the sedimentation, right? So it replaces the gravitational forces. You might have heard that centrifugal force is way too much stronger than gravitational force. In short, centrifugation provides a convenient method of separating either two immiscible liquid or a solid from a liquid. The equipment used for separating are known as centrifuges, right? So please remember this slide. You can pause the video to understand it yourself. Read aloud the definition so that you get a proper idea of it, right? Next is process of centrifugation. Process of centrifugation is very simple. As I mentioned in the last slide, that the centrifuge, that is the equipment, right, is contains of what? Container which contains mixture of solid and liquid or two liquid that are immiscible, and they are rotated at very high speed, right? The mixture is separated into its constituent parts by the action of centrifugal force on their densities. A solid or liquid of higher specific gravity is thrown outward with greater force. Therefore, it is retained at the bottom of the container, leaving a clear supernatant layer of pure liquid. Here, when the mixture is introduced inside the equipment, that is centrifuge, it is separated into two parts, right, by the action of centrifugal force. Why? Because they are made up of, they have actually different densities, right? A solid or liquid of high specific gravity is thrown outward with a greater force. Whoever have the high specific gravity will be thrown outside, right? It's just like a tornado or a twister. Therefore, it is retained at the bottom of the container, leaving a clear supernatant layer of pure liquid. The speed of the centrifuge is commonly expressed in terms of the number of revolutions per minute or of the rotor. So here the basic things are on their densities. This is the most important basic principle of centrifugation. You need to understand that they are separated based on their densities, right? The second thing is revolutions per minute. The speed of centrifuge is commonly expressed in this term, that is, in short, you can also call that RPM, right? So now what we are going to focus is about the applications. The first application is in production of bulk drugs. Generally, centrifugation technique is used to separate crystalline drugs such as aspirin from mother liquor, right? Free flowing product result due to removal of traces of mother liquor and avoidance of any effervescence, right? So in bulk drug production, this is most important whenever you need to separate out crystalline drugs. The second is production of biological products. Most of the protein drugs and macromolecules are present as a colloidal dispersion in water. By these methods or by normal methods, it is very difficult to produce them in large scale, but centrifugal methods are useful for preparation of these type of components. Insulin can be obtained in pure form by selectively precipitating other fractions of protein and subsequently separating them by ultracentrifugation. Centrifugation is employed for separating the blood cells from the blood, right? So this is, as you can see, production of biological product mainly in separating the blood cells from the blood, right? So it holds a very big importance in this industry. Next is biopharmaceutical analysis of the drug. Drug present in the blood, tissue fluids and urine are normally present in the form of colloid dispersion. Centrifugation is used for separating the drugs. This method is essential for the evaluation of pharmacokinetic parameter and bioequivalence studies. So, the most important thing here is analysis of the drugs, right? You can easily perform biopharmaceutical analysis of drug which are present in blood, tissue fluids, urine, etc. Right? So, this method is very important pharmacokinetically and also in bioequivalence studies. Next is evaluation of suspension and emulsions. Centrifugation method is used as a rapid test parameter for the evaluation of suspension and emulsion. In your past semesters, you might have studied about basic about suspension and emulsion, right? Normally creaming is a slow process in emulsion. This process can be fast by you can fast this process by inducing stress condition using a centrifuge, right? Because creaming is slow, you can speed it up with centrifuge. A stable emulsion should not show any sign of separation even after you are centrifuging at 2000 to 3000 rpm, that is revolution per minute at room temperature, right? Next is determination of molecular weight of colloid. Determination of molecular weight of any polymer is not possible by any random or usual methods. Ultracentrifugation methods are used for determination of molecular weight of serum albumin, insulin, methyl cellulose. Centrifugation or ultracentrifugation is also used for ascertaining the degree of homogeneity of the sample. For example, insulin is a monodispersed protein composed of two polypeptide chain, whereas gelatin is found to be polydispersed protein with fractions of molecular weight, that is 10,000 to 1 lakh, right? So basically you can determine the molecular weight of colloids using centrifugation. So, in my next video lecture, we are going to focus the basic, that is what theory of centrifugation holds. This is really important to understand the process of centrifugation and how the other equipments that are centrifuge work. So, please stay connected. Till then, thank you so much for paying attention. Please stay connected to Centrifugation 1.2. Thank you.